Hello.
Yes, very good evening, sir.
Very good evening, sir.
Sir, your voice is echoing. I think there are no devices are running at your end.
Okay, just I'll try to because I joined from two PC. That's why maybe. Just
Okay, okay.
wait.
At one PC, make the volume off the volume. It's echoing us.
Yeah.
Yeah, okay, sir. A very good evening to all. Am I audible to all delegates?
Yes, sir. Audible.
Yes.
Uh, Thank you. Thank you so much. Thank you so much. So again, a warm welcome and very good evening to all dignitaries, organizers, esteemed faculty members, and all delegates presented in today's online session of this 3-month certificate program on research methodology and biostatistics. Today, it is with great pleasure and respect that we introduce our resource person for today's online session, Dr. Mukesh Kumar Naria, sir, a distinguished scholar and researcher who currently serves as the head of the pharmacology at the Institute of Teaching and Research in Ayurveda, ITRA Jamnagar, Gujarat. Sir has an impressive academic background, having completed his M.Pharma and PhD in pharmacology, and he brings with him a remarkable 22 years of teaching and research experience. Dr. Mukesh Kumar sir's exemplary contributions to the field of drug research have been recognized by the Central Council for Research in Ayurvedic Sciences, CCRAS, Ministry of Health, which honored him with the Young Scientist Award in Drug Research. His commitment to education and mentorship is evident from his guidance to 22 M.Pharma Ayurveda students and 14 PhD research scholars, fostering the next generation of pharmacological experts. Throughout his career, Dr. Naria sir has held several significant roles. He has served as the in charge of the CCRAS Institute in Ahmedabad, coordinator of the Department of Project at Gujarat Ayurveda University, member of the Ayurvedic Pharmacopoeia Committee at the Pharmacopoeia Commission for Indian Medicine and Homeopathy, and coordinator of the Technical and Research Cell of IPGTNRA. Additionally, he has coordinated the PG Pharmacy Cell at ITRA. Dr. Naria sir has been recognized as key nominee by the Committee for the Purpose of Control and Supervision of Experiments on Animals, Government of India. His prolific research output includes 95 research papers and 1 patent in India, and he has successfully completed 22 research projects. His expertise and insights have been sought after the numerous conferences and seminars, where he has delivered 29 guest lectures and played pivotal roles in organizing 10 conferences and seminars as a member secretary or coordinator. He is a life member of the Indian Pharmacological Society, reflecting his ongoing commitment to the advancement of pharmacology and his active engagement with the professional community. Today, we are privileged to have Dr. Mukesh Naria sir to share his wealth of knowledge and experience with us. We look forward to session and this with his profound insights and contribution to pharmacology and Ayurveda. Please join me in extending a warm welcome to our respected resource person for today's live session, Dr. Mukesh Naria sir. Please, sir, welcome, sir.
Thank you for your introduction. So I'll start my topic with the basic pharmacology and toxicity related in perspective to the Ayurvedic drugs. Screen. Just wait. Screen is visible or not?
Not right now, sir.
Not right now. Okay, wait.
Now it's still loading. Now screen is visible.
It's visible now?
Yes, visible, sir.
Okay.
Visible, sir.
So already intro given. So I'll start my talk with the basic pharmacology and toxicology, particularly to the highest perspective as a modern context in both way. So all of you know the new drug discovery, that is of conventional drug discovery, that mainly include the start from the target identification, then target validation, hit identification, lead identification, optimization. So mainly two parts include, that is of lead generation and lead optimization. Mainly afterwards, the bottleneck is that part of is the preclinical trials. Preclinical mainly includes start from the in vitro cell-based assay, then in vivo, then clinical trials. So when drug move from the parts of lead optimization to clinical trials, the very important the steps that is called as preclinical trial. So these steps almost includes approximately 10 to 12 years. That is of conventional drug discovery program. But nowadays, mostly in case of reverse pharmacology, that start from clinical to preclinical, that also start with the preclinical trials. So once drug is proved in clinical trials, that again move to the preclinical trials for its scientific documentation and molecular mechanism of action. So all of you know the source of drug mainly include natural, animal source, microorganism, that already proven some of examples that already marketed drugs, but mainly I concentrated that is particularly of recombinant DNA technology. The drugs which are developed through DNA recombinant technology, that is of growth hormone, insulin, erythropoietin, heparin, because the most of the drugs which developed through recombinant DNA technology, previously that drug was obtained from animal source, and the animals sacrifice all over world for the development of such drugs. So nowadays DNA recombinant technology changed the particular part in case of development of such drugs. This is the example of human insulin. You see here human insulin jet, that typical part that inserted to the plasmid loop bacterial DNA. Then human insulin gene, this round shape, that is plasmid loop of bacterial DNA from that recombinant bacterium. Recombinant bacterium, it is developed in fermentation tech, and that recombinant bacterium produced that insulin. That developed the insulin as a first is raw and then purified insulin, and that developed as drug. So such newer technology that changed the particular tradition in case of development of drugs. So another part, that is of reverse pharmacology. That first example I given, the drugs that obtained through plant source or natural source, that can develop through reverse pharmacology. And reverse pharmacology, that first part was that inspired by means of ancient medical science, that is traditional system of medicine. Why? That typically integrated document hits into lead by transdisciplinary exploratory studies. In case of reverse pharmacology, the typical part is safety part. Why? Because efficacy become matter of validation. Efficacy is already proven. So already drug is marketed and already that prescribed by the physician for the particular disease, and already given in the traditional books or ancient science. So here, when you something you extract from the medicinal plant or natural product, the safety become more important starting point than the efficacy. In case of a traditional system, the first three parts, the in blue lines, that is typically for when you develop the drugs. That is of what you call as the drugs when you deductive approach, when you go for the extraction, then isolation, then develop the drugs for clinical trials. The first three is important. But when you go with the traditional system of medicine, because that is particular of holistic in nature. So when you think about a holistic nature, how reverse pharmacology help to the traditional system in the case of scope is to understand the mechanism of action at multiple level, because the major lacuna for the why, because most of drugs not accepted scientifically because of the lack of mechanism of action in terms of modern mechanism to optimize the safety, efficacy and acceptability of leads for the acceptance of drug in the world. So these are the few examples that drugs developed through reverse pharmacology by means of deductive approach. I'll also explain the holistic approach. First, we'll discuss about the deductive approach. You see curcuma longa, curcumin, taxol, taxus baccata, senosites, and many more drugs are there which can develop through reverse pharmacology concept. So when you think about reverse pharmacology, you have to know about the basic concept of pharmacology. Basic concept of pharmacology, that is of two parts. One is pharmacokinetic and another one is pharmacodynamic. All of you know pharmacokinetic and dynamic. I will not go in details in theoretical parts, but I will explain the pharmacokinetic and dynamic, some of the practical aspects that typically require for the development of drugs. All of you know the pharmacokinetic, mainly the absorption, first of transportation, absorption, distribution, then free drugs, particularly that produce its effects, either metabolize then excretion or otherwise bind to the receptor and produces efficacy. So all of you know in case of absorption, the main terminology is the bioavailability, and nowadays two terminologies widely everybody is spoken about, the bioavailability and bioequivalence, that is called as BABE study. In case of modern system of drugs, the BABE study is much more important. In all the CROs, that is contract research organization, then mainly involved in the BABE study. What is bioavailability? That percentage of drug reached to the plasma, that is called as bioavailability. All of you know. But bioequivalence, two formulation of same drug having equal bioavailability, because it doesn't matter that if you prepare some drugs, for example, if you prepare paracetamol, when you administer paracetamol as a oral, when you give in IV or something, if you modify the formulation, its bioavailability is not always same. That's why the dose will vary when you change the what you call is the route of drug administration. That's why 500 milligram of paracetamol is equivalent to 150 milligram of paracetamol when it was given, when it will be given in IV form, and 500 when it given to the oral form. That is the bioequivalence. means, and in another way, when you formulate two, for example, the first part is, you see the drugs, that is, one is generic, that is in pink, and another blue in reference standard. So both drugs, that bioavailability is, so if you see, both are having the almost equivalent bioavailability. Another example, metformin when given as a single constituent, and when it given in formulation concept, almost bioavailability is similar. So that is called as bioequivalence, and you have to determine the bioequivalence for the test drug when it administered in the different route of drug administration. So that is the concept of bioavailability and bioequivalence. So you see here, I'll give typical the charts. When you administered the drugs in intravenous, immediately peak level was observed, and as the time pass on, the bioavailability decrease. Same way, intravenous, the somewhat late, intramuscular somewhat late, then subcutaneous, and then last oral. So this is the difference between the when you administer the drug. So route also start when you change the route of drug administration, when you change the formulation of particular component, then you have to go for the pharmacology, that is called as preclinical as well as clinical. It doesn't mean that the drugs, if it is available in oral form and it's accepted, that you simply change the formulation. No, if you change the formulation, if you change the route of drug administration, then also it requires minimum 200 to 300 crore rupees. Because why? Again, you have to start from the preclinical and clinical. That is a pharmacology and clinical trials. You see, these are the certain changes in the doses form, as the you all of you know the routine doses form, that is tablet, injection, and all this. But nowadays, the few more formulations which are have the novel formulations and widely practiced in the market. You see jet injection, that give the very good particular part in case of what you call is the injection of insulin. Previously, insulin injection, that was with the conventional injection and create lot of uncomfortable to the patient. But nowadays, that injection, you just feel it in the this type of what you call is formulation. You see, this is the conventional needle injection. Simply inject the formulation at particular part. When you inject with the jet injection, it simply what you call is the absorb the drugs at particular in the spreading form. So here I'll give the example. Same way, iontophoresis with the only cathode and anode rays, that is anode and positive and negative charge. With that part, the drug directly absorbed in the palm or particularly in feet. So that what you call is the excessive sweat in case of palm and sweat, that is typical disease. In same case, if you administer the drug through this way. Same way, adhesive unit or patches, that is scopolamine, during motion sickness, the drug loaded in that particular adhesive unit and stick to the particular parts of hand or what you call is leg, that release the drugs for longer duration, and you cannot need the drugs to take at a typical interval. Implants and inserts, estrogen and progesterone, and many hormonal pills. For example, the Maladi you have to take on daily basis. If you administer the drug in such form, it retards the routine uptake of drugs at regular intervals. So these are the certain novel formulations that produce the what you call is that is of delayed release of drugs, sustained release of drug, control, continuous release of drugs. That is different terminology. Ultimate aim of all these formulations is the sustained release, that is release of drug for prolonged period. So you see immediate release, that drugs, if you put it immediate release, the best example is of the drugs you put it under the tongues, it immediate release the drugs within few seconds or within minutes. So delayed release, the drug release over the long period of time, control and sustained release. Here I'll show these some examples. You say glucose phase 500 milligram, and another example is glycomet 850 SR. So glucose phase 500 milligram is conventional dosage form. Immediately drug what you call is the disintegrate, dissolve, and drug directly absorbed as a bioavailable drug and reach to its peak level. But in case of sustained release formulation, you see, in case of sustained release formulation, the two dose you have to include in sustained release. First is of what you call is conventional. Why? The first instant you have to the drug absorbed and reach to its peak level. Once it reaches to its peak level, you have to maintain the drugs for longer duration. You see the graph. This is the conventional means once it reaches to peak level, again comes to down, again you take the second dose, again reach to its peak level, goes down, like that. But in case of sustained release, once you take the drugs, it release the drugs first, the conventional directly reach to its peak level, then release the drugs for around 4 hour, 5 hour, or up to 8 hours, and produce the drugs for the longer duration. So nowadays most of the pain relieving medicine, some of antacids, wherever the in the backside SR is written, SR means sustained release. So one thing you have to very careful, when it was written as SR, means you have to tell to patient, because most of cases the patient sometimes outside they'll just grind the drugs and make it powder. So in case of SR drugs, the physician should advise to the patient, should not grind or should not make powder outside. Otherwise what will happen, immediate 850 milligram of drugs administered at a time, and it may produce certain side effects or adverse effects. So when you administer the, when you prescribe the SR, you have to advise to the patient how to take the drugs. Similar way, distribution of drugs. Just I will concentrate on apparent volume of distribution. Apparent volume of distribution, matlab total amount of drugs in body would have to be uniform distributed to provide concentration of drug actually measured in plasma. Because sometimes when you administer the drugs, it always distribute to other many compartments such as plasma, interstitial fluid compartment, transcellular fluid compartments, then BBB when it cross the BBB, or some other compartments. So distribution it's reduced, and if the effect of drugs only observed with the free form of drugs, if drug bind to the particular proteins, if drug distribute to other compartment, it not produce the effects. And the best example already I given, you see tetracycline to bone, phenobarbital to brain, chlorpromazine to eye, and all of you know the example is sodium warfarin. It has 72 hours of what you call is distribution and excretion from the body. Why? Because now more than 90% of drugs bind to the protein. That's why free form of drug not available to the plasma, and it may not produce the effect, or may sustain for longer duration in the body. And by mistake, if patient take the drugs at regular interval, for example morning and evening, if it will give the cumulative addition of drug in the body and may produce the adverse effects. So you should know the apparent volume of drug distribution when you formulate the formulation. Pro-drug, all of you know the active form of inactive drugs, and best example is dopamine. The L-dopa is pro-drug of dopamine. And in case of Ayurveda or Ayush system of drugs, when you administer the drugs with certain anupan, certain sahapan, it may increase viability, it may decrease the side effects, and many more things. So advantage is increase absorption, elimination of unpleasant taste, decrease toxicity, decrease metabolites. So lot of scope is there. In case of Ayush system of medicine, for the development of pro drugs, because here the single entity when you administer, particularly rus preparation, bhasma preparation, when you administer the single entity, the efficacy may a different one. And if you administer the same drugs with certain anupans or sahapan, the efficacy may change or increase or decrease. So the concept is the ultimately something may change which can either delay toxicity or either increase viability, increase the efficacy, decrease the toxicity, like that. So lot of scope is there in case of pro-drug, development of pro-drugs. Here you see the metabolism of drugs by means of reduction, oxidation, conjugation reaction. And I want to discuss particularly with this picture the enterohepatic circulation. Why? Because certain drugs which can eliminate through what you call as glucuronide conjugation in the intestine through bile, when drugs are excreted through bile. So again in the intestine is very long and particularly responsible for absorption of the all the materials, either nutrients, either drug, anything. Okay, so the what you call is brush border membrane, location of many enzymes of brush border membrane, that is of certain what you call is either absorptive or either what you call is for the digestion purpose. Certain enzymes, I'll enlist sodium ATPase, gamma glutamyl transpeptidase, amylase, disaccharidase, and many more enzymes are there, which either responsible for digestion or absorption. And through this mechanism, the drug also got reabsorbed and again reach to the systematic circulation. From systematic circulation, again go to liver, again that excrete to intestine. So this is circulation, that is enterohepatic circulation. So drug sustained for more duration in the body, and if you administer the another dose of drugs, it may produce the certain side effects or adverse effects. You see the excretion pathway, mainly urine, bile, lungs, saliva, milk, sweat, intestine, and that all that is either through active secretion, passive diffusion. While I'll explain all these things, because when you prepare the drugs, when you formulate the formulation, you should know these all parts. Okay, how drugs get absorbed, how drug get distributed, or at what part it reach to the particular concentration to the particular site, and how interact with the what you call a site or certain proteins or either enzymes or either receptor, and modify the pathophysiological function of the particular disease and produce its efficacy. The therapeutic index, particularly you have to determine in case of which drugs, opioid drugs, highly potential drugs in case of that Basma, Rush Oshadi, that we call as highly potential drugs, because the very low dose of drugs, it produces the very dynamic efficacy. So in that case, you should know the drug safety, you should know the therapeutic index. Therapeutic index means how much dose should produce the efficacy and how much dose should produce the toxic. So between these two parts, you see the black line and red line, the between this part is the efficacy of drugs. The more difference between therapeutic effects and toxic side effects, the dose is the determine the safety of drugs. For example, the 50 milligram is the therapeutic efficacy dose, and if 500 is the toxic dose, the 10 times is the therapeutic index. 10 times. Why? Because the less the difference between the dose which are responsible for therapeutic efficacy and safety is much as the, and one is not preferable at all, because ultimately LD50 divided by ED50, that is called as therapeutic index. You see here I have mentioned some of the drugs which have narrow therapeutic index and wide therapeutic. This is of modern, and if you think about the Ayush system, narrow therapeutic index drugs of all the wish and wish drug mentioned, such as Langley, then what you call as Aconitum, and all the drugs which are mentioned in the Schedule J, and Schedule I or J, yes, Schedule J. So all the drugs which are mentioned under Schedule J, that is of narrow therapeutic index. And in case of modern system of medicine, digoxin, warfarin, lithium, that all drugs are narrow therapeutic index. And wide therapeutic index in case of Ayush system of medicine, all the herbal drugs, for example, amla, that all drugs, what we call as the churna, we are using mostly 3 gram to 6 gram. If you administered even 10 times more than that of therapeutic dose, then also it may not produce the toxic effects. So that type of drug have wide therapeutic index. So here I mentioned why the failure of drugs during clinical trials. Here I have some mentioned. You see, many persons asked me, sir, whether the drugs pass from preclinical trial, that same effects may observe in clinical trials. No, that even you see preclinical trials, clinical trial, phase 1, phase 2, phase 3, that all are predictive. Predictive. Why we call this predictive? Because when drug move from preclinical to clinical, 90% drug fail. Only 10% of drug pass to the phase 1. Why? Because preclinical, that is the prediction in either in cell-based, either in what we call as the computer-based model, either through animal models, then drug move to the clinical trial. But it doesn't mean that you have to start the drug directly phase 1 trials. So even I listed here some that I download from the FDA sites. You see, phase 1 trial, phase 2 trial, also there is chances that even drugs marketed drugs also there is chances of failure. Here in phase 1, you see that due to safety reason, 30% of drug fail. Efficacy reason, almost 28% drugs fail. Even phase 1 trial drugs successfully completed. When moved to the phase 2 trials, almost 60% drug fail. Even drug move from phase 2 to the phase 3, almost 30 to 40% drug fail. Phase 3 to even marketing approval, that is called as NDA, new drug application. When it move, the almost 20 to 25% drugs fail. And even after marketing, all of you know the examples of nimesulides. Even after long history use in clinical trials, now clinical phase, and now it withdrawn from the market. So this is the drug failure chances, and that's why the more people and industry move through the reverse pharmacology concept and drug repurposing concept. I'll come to drug repurposing concept. And you see, I enlisted drug approval from 1997 to 2021. Very low approval, low chemical entity of drugs or biological approval by FDA. Here you see not even close to 50. And during 2018, almost biologicals, due to increase in biologicals, the number is to 59. But almost, if you see, this is the data worldwide, and worldwide the 50 chemical entity per year is very low. So that's why because very low, the success ratio is very low when you move to the conventional drug discovery, and that's why more people are concentrate on drug that is developed through reverse pharmacology and drug repurposing. The drug repurposing, all of you know, during COVID time, many eyes drug repurpose, and even modern drug also repurposed for corona. The best example is chloroquine. Previously, they all thought that chloroquine may hit to the targets in virus, but after all, it was found that it was not that much of efficacious. And in modern system of medicine, all of you know the example is folic acid, then methotrexate. Nowadays, methotrexate, previously it was sanctioned for anti-cancer drugs, but nowadays it's choice of drugs for anti-arthritic and even to the autoimmune disorders. Folic acid, nowadays then same way, many more drugs. That drug repurposed, previously it was approved for some another purpose, then with the certain approach. Aspirin also example of drug repurposing. Previously it was for analgesic activity, but nowadays it was used as antiplatelets. So what is drug repurposing approach? And finding new uses or indication of existing drugs or formulation. Drug repositioning approach for emerging or re-emerging infectious disease. In drug approval by FDA, 30% are of repositioned drugs. And nowadays, all of you know the what we call as the ashwagandha. Ashwagandha for some other as a what we call as the brain CNS purpose, no, it's widely used. But in case of, if you see the use of ashwagandha in ancient science of system of medicine, it mainly concentrate on what we call as the Rashayan or other reproductive related issues. In drug approval for orphaned or difficult to treat disease, mainly drug repositioning, the non-clinical development of drugs. Drug repositioning for the preclinical trials, it mainly includes CMC. CMC matlab chemistry, manufacturing and control. Animal studies mainly include safety and efficacy. Clinical pharmacology, that is phase 1 and phase 2. So from the clinical pharmacology, you get the idea about the doses form, how much dose it may produce, the efficacy, safety trials, that is in phase 1 and phase 2. So clinical pharmacology mainly concentrate on phase 1 and phase 2, while preclinical pharmacology mainly concentrate on animal studies, that is safety and efficacy. So overall, if you see drug repositioning, it start from in silico screening, molecular docking, pharmacophore modeling, that is of computer-based drug design. Then disease and therapy. When you move to the preclinical, it includes the in vitro assay, in vitro cell-based assay, in vitro cell line studies. In vivo tests include mainly the preclinical trials in animal models, and experimental screening, that is of HTS, high throughput screening, and at last preclinical safety and efficacy and clinical safety and efficacy. So nowadays, all of you know the molecular modeling, that is in silico and network pharmacology. In silico, through in silico, many drugs develop, particularly in case of modern system of medicine. But when you thought about this traditional system of medicine or what we call as the ancient system of medicine, because that particularly include the holistic approach. In case of holistic approach, in silico approach may not that much useful, but that in silico approach, when you convert to the network pharmacology, it give very good insight into the molecular mechanism of drugs for particular disease or particular mechanism of action, and emerging techniques for natural product, drug discovery and scientific approach. Molecular network by integrating multidisciplinary biology, including bioinformatics, system biology. It based on multi-targeting potential of effective drugs as well as multi-ligand SIGAN target approach. You see, in case of the plant-based formulation or these holistic drugs, here multiple constituent, multiple phytoconstituent, multiple phytoconstituent, multiple proteins, multiple targets. So not a single targets. And in case of modern system, single drug, single target. But here multi-ligand, single target, or multi-ligand, multi-targets. So the as a whole, the particular network will or network can you can plot, and at last you can find out the what are the probable mechanism of action or probable the molecular mechanism of your drugs. The disease involve interaction of multiple genes. It detect how and where in disease network target and ligands can act. So ultimately, network pharmacology that start with the whole plant, chemical database, then therapeutic activity, potential components, potential target proteins, cytoscape, component target protein, and at last this type of protein target protein pathways network was obtained. Particularly, I'll start this type of approach since 5-6 years back before COVID time, and we got very good results when you start the start the particular plant or what the any formulation. If you include this approach along with preclinical trials. Single approach may not be useful, as already I told previously. Single approach may not useful to find out the whole mechanism of traditional system. You have to follow the multiple approach. Multiple approach means start from in silico, network, then cell-based approach, in vitro approach, preclinical trials. So if you include all these approach, then you get something for the molecular dynamics or molecular mechanism. So you see, these are the parts of what we call is the ligands or the targets, the potential targets. Potential targets, either receptors, either proteins, either this is called as pharmacodynamics. How drug act, what drug does to the body, that is called as molecular or pharmacodynamic. And pharmacodynamic includes this is if drug bind to the at the molecular level through receptor. So you see, these are the some enlisted receptors, that is called ligand gated ion channel, G protein coupled receptor, kinase link receptor, nuclear receptor. Did know any typical what we call is the full formulation or has a plant based drug that typically identified for this type of molecular mechanism. The example of that is of GABA receptor. You see, all the CNS drug, particularly anti-anxiety drugs, anti-conversion drugs that act through this receptor. This is the example, typical paucept, alpha, beta, and gamma units of GABA receptor. Once it open the channels, chloride channels rush to inside, and hyperpolarization of cell and resting potential decrease to the minus 120. That's why it's not easy to again stimulate that particular cells. The example is the GABA receptor, that chloride channels, and the drugs act through that receptor is diazepam, barbiturates, and many more of Ayurvedic drugs that particularly calm the use as a madhya may act through this type of receptors. But you have to find out and document the and scientific study for the whether that drug bind to receptors and what is the basis of the madhya rasayan. Same way, enzyme or kinase link receptor, once drug bind to the extra domain of that particular receptor, it may dimerize that autophosphorylation. Once it goes autophosphorylation, the RAS family, the GTP converted to GDP, RAF, MEK, MEF kinase, and various transcription and produce the efficacy in the nucleus. Same way, the steroid binding domain, the only receptors which can produce, that is called as nuclear receptors, and through nuclear receptor that directly produce the efficacy with the binding of nuclear, that directly control over the cell activity, that is through transcription and translation, all of the protein synthesis, that is through phosphorylation, dephosphorylation. So if drug act through this nuclear receptor, it directly get entry to the cells. And you see many the insulin many receptor based disease, receptor based matlab change in the what you call is the pathophysiology produced, and that is examples, that is myasthenia gravis, insulin resistance diabetes, testicular feminization, then hypercholesteremia, cardiomyopathy. The example of nuclear receptor, vitamin D and steroids, mostly drug act through this. So preclinical drug development mainly includes the drug safety and efficacy profile, genotoxicity, reproductive, carcinogenic study to predict the drug behavior in human in clinical trials, how drug behavior, what is the drug tentative efficacy, what is the tentative side effects, and what is the predictive behavior in humans. That's why I told the preclinical is the predictive behavior. So mainly it includes the animal models that include mouse, rat, guinea pig, rabbits, large animals, dog, more likely monkey. And in India, CCSC, that is Committee for Control and Supervision on Experiments on Animal. If you want to go for the animal house approval, the CCSC give the registration for animal house. Previously it was CPCSE, but nowadays it was changed to CCSE. And IAC, Institutional Animal Ethics Committee, for approval of small animals. IAC for Institutional Animal Ethical Committee. Same way for clinical trial, IEC, Institutional Ethics Committee. The Institutional Animal Ethics Committee have power to approve the experiments on small animals. But in case of large animals, if you want to approval for large animals, IAC is the recommending authority and CCSD is the approval authority. CC is the headquarters at New Delhi. So that have power for the approval of large animals experiments. So Ayurvedic drug optimization, refining drug formulation, and when you go for the refining of drug formulation, at least you have to start from preclinical trials, because when you change the number of put, when you change the something, that you have to see that again I'll come to the IUS 170 guideline. When you change something, you have to start with the preclinical trials. Formulation substance sometimes may exhibit different biological activity, and nowadays phytopharmaceuticals, because mostly Ayurveda industry that use the either extract based formulation or either change the certain formulation, either change the method of preparation, then you have to start with the preclinical trials. With Anupan and Sahapan, as already I explained, the different biological expression, because with one Anupan, the expression may change, and another with the Sahapan concept also, particularly with the metal and mineral preparation. Sahapan have very great a very what you call is the vast concept in case of Ayurveda. Many drugs prescribed with Sahapan. To improve its drug characteristics, develop mechanism-based pro-drugs, and much more important is stability of drugs. Stability of drugs, when you formulate something, when you change the formulation, stability, you have to see the viability, and you have to see the pharmacodynamic profile. Just I'll give the example. In our lab, we carried out very detailed study on opiumcia. You all of you know that is called as Atlathor in the Saurashtra region, and very beautiful fruit that is radish. You see here picture, if you see this radish fruit. So previously the जो शेफर्ड जो गाय भैंस वो चराते थे, एंड सौराष्ट्र रीजन में पानी नहीं मिलता था, जब बाहर चराने के लिए पूरे दिन बाहर रहते थे, तो देन दे यूजुअली टेक दिस टाइप ऑफ फ्रूट्स एज ए न्यूट्रिटिव पर्पस, एंड अगर उसको आप ऊपर का छिलका निकाल के खाएंगे, दैट इज हाईली व्हाट यू कॉल एज द, मतलब टेस्ट में भी बहुत बढ़िया है, न्यूट्रिटिव पर्पस भी है, और इट कंसिस्ट हाई कंटेंट ऑफ वाटर. सो प्रीवियसली, एंड खास करके जो विलेज पीपल है, दे routinely use the fruit of this shwaras of fruit of this, particularly in anemia, and in case of pregnant lady, they are routinely used. So we took the detailed project on opiumcia, Professor R.N. Acharya previously he was in ITRA and now he's the DG of CCRAS. Both of us, we started the preclinical and clinical trial. He took many projects from on this plant, and I look after the preclinical trial on this plant. And you see many articles also published, hemolytic anemia, we see the example. And recently we completed the work on reproductive and development safety evaluation. Why? Because this particularly prescribed in pregnant woman. And one more detailed project again Acharya started on cancer patient, because most of chemotherapy, the side effects of chemotherapy is the anemia. That's why if along with the chemotherapy or along with the cytotoxic drug, if you prescribe such the use of these drugs, whether it revert the or with whether it increase the quality of patient or not. So many the work we completed. And one more drug, that is of Catupilla sagrinigal leucopyrus, that also we studied in very what you call is the vast part that includes start from the preclinical. It start from the first, we start from the chemistry part, then CMC part, then preclinical, then clinical. And clinical also almost more than 800-900% they completed. And preclinical part I look after by me in the animal trials, and clinical parts Professor Dudmal sir. So he's the head of the Salia department. Many works he completed on these plants, and many articles also published. We also published the monograph on Saccharonegal leucopyrus. Previously, it was found that these plants brought by one Sri Lankan scholars from Sri Lanka, and afterward it was found that this plant is widely available in Saurashtra region of Gujarat also. And nowadays, the many projects going with the even modern hospitals, and they send the patient of this what we call is the diabetic wound, and we got very good results for these plants. So very detailed study was completed on Saccharonegal. So these are the few examples we completed in for the preclinical and clinical both. So it includes pharmacology and clinical both. So molecular markers we are missing in particularly in clinical and preclinical. Why? Because these all molecular markers are very costly, and mostly while doing the research studies we miss these markers in our study. Why? Because once you see, when you start the toxicity study, when you start the efficacy study, you already doing lot of work throughout the years, and at last when you withdraw the plasma, or when you take the certain what you call is the tissue for either histopath study or either for the certain highly sophisticated this molecular marker or instrumental techniques, we miss this part. So at least you have to include certain markers in your study for the molecular mechanism of drugs. You see adiponectin, leptin, HOMA-IR index, inflammatory markers. Inflammatory markers nowadays lot of importance after COVID. Previously, there was no such markers they included in particular studies, but nowadays TNF alpha, IL, IL-6, this may require the either what you call is the ELISA or either RT-PCR. You can go with that studies. Oxidized LDL. Oxidized LDL is the marker of lipoprotein, mainly including oxidative stress. When you go with the anti-diabetic study, when you go with the atherosclerotic part or either cardioprotective activity, at least you have to include oxidized LDL. And oxidized LDL nowadays much more important molecular marker for the find out the importance of drugs. Same way ghrelin, cholecystokine, both hormones. Now we are including in our deepen pachan activity. Previously we are not we also not included. Previously we done the deepen pachan activity with the certain enzymes, all the gastric enzymes such as renin, then other parts as a mucosaccharides, that is lipase, then trypsin, chymotrypsin, disaccharidase, and peptic activity. But nowadays we also include ghrelin and cholecystokinin. Very good results observed with Trico2 formulations and many more Ayurvedic drugs. Same way GIP, glucagon like peptide 1, then anti-Mullerian hormone, gut microbiota, GLUT2 and GLUT4. Particularly this nowadays a very interesting targets for anti-diabetic drugs. Then damage associated molecular patterns, and many more. You see kidney injury marker, LDH, GLDH, HDH. Recently we done the work on Anand virus. Anand virus for its safety and efficacy. Safety in terms of its oral toxicity study, then genotoxicity study, then antipyretic study, and in that part almost all the molecular marker we included, whether drug produce any changes in such molecular marker at the end point. So the many inflammatory markers, then LDH, SDH, almost all came, then daily drug-induced liver injury. So this at least you have to include in your study to find out the molecular mechanism of your drugs. Now I move to the toxicity studies. First, previous parts already I explained, that is of efficacy study, and this to the toxicity study. Why toxicity study? Assessment of safety of Ayush drugs, define possible adverse effects, and you see everything either in lower or either in here, either just you see green part water, then caffeine, and another is the poison. You see water 90,000 mg, 90 liter almost, caffeine 250 mg per kg, and highly toxic plants that is of 2 mg per kg. The difference is only in the dose duration, but if you not take the things or either drugs or formulation not in proper dose, not in proper time, it may produce the side effects, adverse effect or toxic effects. So it is must to find out the therapeutic index. What is therapeutic index? That already I explained. So therapeutic index, to find out the therapeutic index is much more important when you work with the highly potential formulation or drug. For potential matlab all the ras aushadhi, all the poisonous or what you call is ovish and upish drugs which require southern parts, which require the maran, amritikaran, these procedures, at least you have to find out the therapeutic index. And another parts you see many more, the western part they publish such reports. The Ayurvedic herbal drug consists of many heavy metals and aflatoxins. That's why it may produce toxic effects, side effects. So you have to counterpart with the study of safety trials, the toxicity study. And one more thing, guduchi. You see identification if you issue the guduchi and one more plant that is called as Cryptostagia grandiflora, almost looking similar. And previously it was incidence in Jamnagar. The 2% in place of guduchi, they took this rubber well, and serious one immediately death, and second that survived, but almost 1 month in ICU, and afterwards cardiac issues, because that Cryptostagia grandiflora produce hyperkalemia, and hyperkalemia is it's very dangerous to the heart. So intermittent cardiac blockage, cardiac block. So you see, sometimes this type of issue also may create so serious what you call is the mislead to the population, because people don't know okay what is guduchi, and reports if published ki guduchi produced this one, but ultimately it was not guduchi but something else. So for the safety assessment, the guideline, one guideline is the guideline for evaluation of Ayurveda, Siddha, and Unani drugs and other traditional medicine of India. That is 170 guidelines. Then Schedule Y, the Schedule Y also give the preclinical and clinical trials. Same way OECD, guideline for testing of chemicals. Then WHO guideline. Then CCRAS guideline. CCRAS, they not formulates themselves, but that is the abstract of other guidelines, and they formulate the guideline for evaluation of drugs for Ayush products. So CCRS also publish the guidelines, CDSO also publish the guidelines. So these are the all guidelines are the what you call is the authoritative published guidelines. So when you proceed for the safety trials, at least you have to refer these all guidelines, and accordingly you have to design your drugs. This is 170 guidelines, as already I told. Medicine based on extract of medicinal plants. This RRR matlab required. When aqueous extract matlab not required. If again there is star mark matlab if required. So if you want to go in detail, you just follow the 170 guidelines. The detailed studies was given when your preclinical trials require, when safety data is required, when clinical trial is required. So here the medicine based on extract 100% requirement is there for the preclinical and clinical trials. So when you go for the toxicity study, require relatively large amount of compound. Purity of compound needs to be very high. Otherwise, impurity may produce the toxicity. And you thought that your drug may have side effects, but the side effects may be due to other reason. Toxicity profile may be documented in IND if you use the ingredients other than the your toxic drugs. So mostly in case of IU's drugs, oral toxicity study that is of acute or repeated dose, dermal, eye irritation, skin sensitization, inhalation, because mostly Ayurvedic drugs administered through oral or some other routes, but not through IV or other thing. When you think about modern drugs, many toxicity studies are there, but you think about the IU's drugs, so only few toxicity study requires, and nowadays, particularly for Bhasma preparation and schedule J drugs, mutagenicity, carcinogenicity, and reproductive. As already I explained, we also conducted reproductive toxicity for many drugs, and mutagenicity for almost 7 Bhasma we completed for its what you call is the genotoxic profile. Genotoxic profile मतलब कि either changes in what you call as sperm abnormality or chromosomes abnormality, that is through chromosomal aberration assay, in vivo micronuclei assay. Many study we completed for Bhasma. So almost 3 to 4 publication out for the such type of study. So duration of toxicity study, how you decide the duration of toxicity study? When drug administered through less than 1 week, then toxicity study may to conduct from 2 weeks to 1 month. When repeated administration between 1 week to 4 weeks, the duration may be 4 weeks to 3 months. Repeated administration between 1 to 6 months, the duration to be 3 to 6 months. Long term 9 to 12 months, and even now CDS show that give to the 24 months, so almost 2 years, particularly for carcinogenic study. Dose conversion, you see, when you want to directly convert the dose, as already mentioned in classical books, you directly convert the dose as per dose conversion ratio that already given by means of Paget and Barn table, that is 0.00264 for mouse, 0.018 for rat. And when you apply the dermal application, approximately 10% of body surface area ratio of the animals. So you have, if drug is there already, then you can design the drug, design the dose of doses from as a TD, TD5, TD10 in case of toxicity study. So acute toxicity at a once, and particularly at higher dose, when single oral dose, when you predict that drug is not that much of potential or not toxic, you directly do the acute toxicity as 2000 mg per kg. That is called as limit dose, and that test is called as limit test. Directly you conduct. And if you think about that drug is toxic at 2000, you can start at different dose level 5, 50, 300, and 2000, and see the observation, that is behavioral changes and respiratory circulatory, as much as parameter you can observe for toxicity trials. In case of 28 days repeated dose toxicity study, number of animals 10 per group, 5 male, 5 female. In case of 90 days, that is 408, 10 male, 10 female, 20 animals. And limit test you can directly start at the 1000, or otherwise you can include minimum 3 dose level of drugs to check at different dose level. Okay, what dose level is safe, and what is which dose is novel, no observed effect, adverse effect dose level. So novel you have to find out, that is therapeutic index. Observation, as many as parameter you can include in toxicity study. As already I give the example of Anand Bhairavaraj. You see, this is the minimum parameters you have to include in toxicity trials, and other examples I already explained in molecular markers. So these are the certain other study or guidelines, that is mutagenicity study. These OECD guidelines for testing of chemicals. Here are there some guidelines you have to follow for the drugs. JAMA in 2008, 2007 or 8, that published one of article, and at that time many drugs banned in Western country. Our lab also received these 5 drugs for its detailed toxicity trials, and we submitted the reports. Previously it was coded drugs, and that it afterwards it decoded as Ma Lakshmi Vilas Ras with gold, Ma Sudarshan Ganvati, Ma Yograj Guglu, Ma Ratna Ras, like that. So these are the examples, and here you see many drugs of very potential drugs. We carried out the toxicity trials in our laboratory, and these are the few published literature on toxicity. And what are the concept of Sodhan, what are the concept of Maran, and put preparation, put matlab different puts after giving the different puts, so that the changes. So you see one of the concept also the Sodir, then Tamra Basma, that incinerated copper with or without Amritikaran, then acute and Rashmanikya with the certain what you call as Sahapan, Anupan, assessment of genotoxicity profile is already I told Rashmanikya. And recently we completed a detailed study on Nag Basma with the different put level, 7 put, 10 put, 30 put, and detailed preclinical trial, oral toxicity, genotoxicity, anti-hyperglycemic, anti-diabetic, clinical trials almost completed. That was started in the Rasashastra and Bhaishya Kalpana department by Professor B.J. Padgiri sir, and he was look after all the drug preparation part and clinical trial, and I was look after the preclinical parts. And at last the detailed study was published on Nagbasma. So these are contribution of our institute for the Nagbasma, and many more contribution from other scholars from the Department of Rasashastra and Bhaishya Kalpana. So here I complete my talk on preclinical part, that is of pharmacology and safety trials, particularly that includes the efficacy and the safety. So when you think about the modern side, you have to design your study accordingly. When you think about the traditional system, you have to design your study accordingly. So both have their importance, both they have different methodologies, but ultimately the evaluation part is almost same. You have to modify the methods according to your drug design, according to the fund requirement, because ultimately preclinical and clinical trial require a lot of funds. When you design with the certain molecular marker, really it requires a lot of funds for the preclinical and clinical trials. So you have to design your work accordingly, with the inclusion of Anupan, with the inclusion of Sahapan, and number of animals, number of subjects, inclusion criteria, exclusion criteria, and many more things. I think all of you know this part. So this is the some of my experience I shared with you. So thank you all.
Thank you so much, sir, for delivering the wonderful session. And now, as usual, I request to all delegates, if you have any doubts regarding today's session, you are free to ask. I allowed everyone to unmute their end. You can write your query, you can directly ask your query.
Actually, one question is there, sir, is there any scope of network pharmacology of Rush Aushadhi formulation? May or may not contain herbal ingredients. Rush Aushadhi also, if you know the last constituent of Rush Aushadhi, last constituent means it required drug characterization. If you go with the drug characterization with the Rush Aushadhi, and if you know the ingredients, okay, what are the last form of that particular Rush Aushadhi, then it chances is there you can go for the in silico as well as network pharmacology, because ultimately you should know the constituent of that part. If you know the constituent, then only it can correlate with the particular targets.
Another query from Karnam Chandrasekhar. Is Guruchi absolutely free for ADR?
You see here another part, is Guruchi absolutely free of ADR? What is your take? Kindly explain, sir. You see, Guruchi, many formulations already in clinical part, and preclinical lot of work published on Guruchi. But that is of Tinospora codifolia, Tinospora malabarica, and many more, even what you call as the different synonyms of Guruchi, and many more species of Guruchi are there. But when you think of Guruchi, as per ancient text matlab what is described either as a either what you call as it's a powder form or other form of guduchi, then make not that much of toxic or not producing any side effects or adverse drug reaction. But if you change the species, you see many the already the published part that is of guduchi, some other species of guduchi, and already I told the the examples of misunderstanding of guduchi in place of rubber well. You see rubber well and guduchi almost similar part, and in Jamnagar both well jise bolte na, both are shrub or climbers, so almost both are climbers same in the one plant, and that's why it must misunderstood by the two person as already explained. That's why it's a or and otherwise guduchi no any adverse effect or toxic effects reported on guduchi as a whole. But when you isolate something from guduchi, isolate matlab pure constituent, then at least you have to move for the safety study. Because when you isolate something, for example, if you isolate morphine from ibuprofen, morphine is single constituent. If you isolate piperine from piper longum, then piperine is single constituent. Then at least you should go for the safety trials. Another part, repurposed drug. Repurposed, when any drugs prescribed for something, again I'll share something. Just wait. Another, if any other question is there, then we'll move to the that part. How many minimum number of animals to be taken in each group for experimental study, non-toxic formulation or drug? That depends on study. Minimum 6 animal for efficacy study, because it gives the statistical significant result. And if you go for the safety trials, if you go for the regulatory requirements, then then you have to follow strictly as per guideline. Nowadays you see, just recently CDSO guideline published 15 animals to 30 animals per group. It is too much per group. If you take 5 groups, it almost 120 to 150 animals per study, and 120 to 150 study for animals is too costly, even lot of manpower requirement. And that's why, as already I told, clinical and preclinical always costly if you include the all the parameters, and that's why. But ultimately, if you go for the academic work or only for the scientific documentation, then you go with the 6 animal per group, and it's worldwide accepted. And almost article, if you refer in all the reputed journals, even in the impact factor journal, Science Direct or PubMed, that almost they publish the work with 6 animal per group. And another, are Rashoshadi giving action via binding to receptor or cross BBB? Are there any pharmacological? No, till that no any Rashoshadi, till that not any evaluated, because that ultimately already I told these are very difficult to find out, because these almost take lot of efforts to go for all these studies. And just I'll share one more, just 2 minutes I'll share. If you want to go for such study, just I'll share. Just wait.
Sir, hello sir.
Just I'll complete this one. Then I'll wait 2 minutes. Wait. Just I'll give you some examples of this highly sophisticated instrument requirement for evaluation of such a study. Just wait. I'll share you. Screen is visible?
It's a white, sir. White screen is visible only.
It's visible, na?
Only white
Screen is visible,
black
but
screen.
it is white.
Okay. You see, these are the highly sophisticated techniques: PET scan, SPECT scan, imaging techniques, CT scan. Through these techniques, you can identify your drug. You see the examples of rat. Tumor localization. This is single rates after 96. How much this is? But these are highly sophisticated techniques, and it require the instruments which almost cost more than 2 crore, 5 crores like that. So many institute in India having such instrument facility. Here you see the I given the example. So when you want to identify the exact mechanism of action of drugs, you that drugs you have to what you call as radio label and then administered to the particular animals, and then you can scan through such sophisticated instruments. You see biophotonic image of single mouse infected with interperitoneal bioluminescence. So with this technique, you can find out molecular imaging techniques. You find out the in vivo drug action, pharmacokinetic profile, biodistribution, mode of action, receptor occupancy, pharmacodynamic profile, biomarkers. So this is the techniques you can, if you want to go for the typical mechanism of drug action, these techniques help you for the such study. And again, as already I told, this is highly costly. Even if you purchase the instruments, that is of 3 crore, 5 crore per year, AMC is almost 15 to 20 lakh rupees, and even dye and drugs and these all are highly expertise things required. So if you want to go for bhasma preparation, when it get absorbed, when it move to the other part, you just biolabel that bhasma and administer to the animals or either to human if clinical trials allow. Then you can then go with such techniques and find out bioactive or MR contrast agents. So these are the techniques again may helpful for the, as already I told na sir, whether bhasma preparation, Rasosades, and find out the whether it cross BBB or not, whether it reached to particular part or not, like that. Okay. Any just somebody asked me again, please.
Hello, sir. I was the one who asked regarding the first question regarding the scope of network pharmacology. So, by characterization, are you aiming at XRD, XRF?
No, not only XRD, SRF. That may not enough, because nowadays, again, if I'll move to the instrument part, you have to take the help of the what you call as chemist, because sometimes only with the instrument part completion of or characterization it may not help to the, because particularly most of in academic institute they send the this formulation to IIT or some research center for their evaluation, but what are the results, the interpretation of particular results is highly solicit give the exact picture. So only XRD, XRF, sometimes without knowing the importance of the instruments, you just send the sample to the research center and they'll give you the results, may not solve your purpose or fulfill your purpose. So at least you have to collaborate with the experts in IIT or in some NIPERS or some what you call as the or CSR institute, and then you may collaborate with them and you just understand what nature of your drug, what the solvent system require, what the instruments may help to your characterization part. So in that way, if you proceed now, then 100% result will get. So for that you have to take the expertise help from the phytochemistry, chemistry part, then medicinal plant chemist, medicinal plants chemist, like that.
So what stream we need to focus on in IIT and NIPER?
Yeah.
What faculty, what stream of faculty should we focus upon while collaborating?
Chemistry, mainly chemistry. Medicinal chemistry, chemistry, organic. They know the, because ultimately structure, the organic chemist or inorganic chemist, they know the things. So you see, I think in Kerala, the Kerala or in CSIR lab, I think Mysore or somewhere, they typically started the lab for the bhasma preparation. If I got the number, you just contact me later on, I'll share with you. So they started the work on bhasma typically, what are the constituent, what are the characterization required for bhasma, and they done very good work, and till that many bhasma they completed. So if I'll get the number from him, I'll share with you. Okay.
Okay, sir. Kindly do so. Thank you. And sir, the second question I need to ask is: are there any animal models available regarding parental dosage forms, especially in the brain activity, to check the brain activity?
What
Any parental dosage form,
your
mice
voice?
models or rat models?
No, your voice is cracked, not that much audible. I understand
Okay.
deep brain and
Sir, I was asking regarding parental models, parental dosage form models, especially to
okay,
check the brain activity. Is there any
okay.
rat or mice models available?
Yeah, many for CNS activity, many models are there. But which type of work you want to initiate? Because in CNS you see anti-anxiety, anti-Parkinson, anti-depression, sedative, hypnotic, what you call as muscle relaxation. Then many more models are there. Which part you want to work
Sir,
specifically?
regarding
Which so
coma,
eh?
coma models.
Coma मतलब which neurotransmitter you want. Otherwise you can just contact me. I will explain you. You just share me your thoughts, then I'll help you for the design of models. Because what you tell, that is very not pinpoint.
Sure,
You
sir.
have to give me some
I'll contact.
design matlab which type of drug, or how you administered, in which disease condition, in which part of brain, which neurotransmitter. So uh, I think if you give me the full detail, either you send me through mail or you can contact me
Yeah.
on phone also.
Dr. Krishnaveni, please ping me after the session. I will share the detail of a mail ID of sir, and you can share the details. Sir will guide you. So, any other query?
Thank you, Amit.
Yes, sir. I have a question, sir. Sir, animal models we are using nowadays for Ayurvedic medicine, are there any
Yes.
limitations because the major concept of Ayurveda, like three dos panch mahabhut and Jat, especially the concept of Agni, where
Hmm.
when we see the effect in the via Dravya Gun, the pharmacodynamics in term of Dravya Gun, so
Okay.
we are getting effect via Ras Gun, Virya Vipak, and then Prabhav. But
Yeah.
in an animal model, we are inducing that particular disease firstly and then treating. So, are there any limitations, or we can develop Ayurveda-based animal model like in
हाँ,
Ayurveda? Acharya Karanga.
एक्चुअली इसलिए मैंने आपको समझाया था जब वो मोलिकुलर की बात कर रहा था ना मैं, तब एक्चुअली आप देखिए एज एज सच हमको अगर ह्यूमन में भी आप त्रिदोष को मेजर करेंगे तो पॉसिबल नहीं होगा. एग्जैक्ट मेजरमेंट तो कुछ है नहीं, ऑब्जेक्टिवली मेजर करना इट्स वेरी डिफिकल्ट. ओके, दैट इज ऑफ सब्जेक्टिव पार्ट. ओके, तो आप कोरिलेट करना है किसी से भी, इवन आप क्लिनिकल ट्रायल करेंगे तो भी आप कोरिलेट करेंगे किसी के साथ. तो लिमिटेशंस क्या है कि कोरिलेशन इज वेरी डिफिकल्ट. फर्स्ट लिमिटेशन वो है. और अगर आप कोरिलेशन कर पाते हैं कि फॉर एग्जांपल कि आप दीपन पाचन एक्टिविटी का सोचे कि दीपन पाचन एक्टिविटी किससे आएगा, तो फिजियोलॉजी में हमें पता है, फिजियोलॉजी वी नो कि स्टमक कंसिस्ट ऑफ gastric juice and it consists of mucus, then peptic and renin and many more other carbohydrates, and intestinal part contain trypsin, chymotrypsin, then breast border membrane consists of ATPase, then gamma glandular trypsin. That all are measurable. Okay, and one more thing I already told, the hormones ultimately responsible for either Dipan or Pachan activity, as already I told, cholecystokinin and these parts. Okay, just I'll give you example. बाद में सबसे आप ऐसा थोड़ा कोरिलेशन कर सकते हैं, बट अल्टीमेटली आपको प्रेडिक्ट, ये जैसे मैंने बोला कि ये सारा प्रेडिक्शन मॉडल है, तो आपको मॉडल तो उसके अकॉर्डिंग बनाना पड़ेगा, आपको पैरामीटर्स उसके हिसाब से डिजाइन करने पड़ेंगे कि विच फुलफिल योर रिक्वायरमेंट्स. फुलफिल योर रिक्वायरमेंट्स मतलब आप क्या देखना चाहते हैं. जैसे भी ब्रेन में भी मैं बात कर रहा था, तो वही डिस्कस कर रहा था. ब्रेन एक कोई सिंगल नहीं है, उसमें भी बहुत सारे न्यूरोट्रांसमीटर्स है, कहां से क्या पार्ट हो रहा है, आप ड्रग देने के बाद क्या मॉडिफाई हो रहा है, क्या नव कंडक्शन चेंज हो रहा है कि क्या हो रहा है. तो ऐसा अगर आप कुछ कोरिलेशन करके करेंगे ना, तो लिमिटेशंस कम हो जाएंगे आपके, और एटलीस्ट यू गेट द साइंटिफिक वे फॉर द. जैसे त्रिकटु का मैंने बोला कि त्रिकटु है, दीपन भी है, पाचन भी है, फॉर एग्जांपल, ओके, और मोटिलिटी को चेंज करता है। तो आप उस हिसाब से, जैसे हमने बहुत सारे मॉडल्स को डेवलप किया, इवन रिसेंटली स्पोर्ट्स पर्सन के लिए यहाँ पे एक ड्रग बनाए थे। उससे पहले पंच महाभूत के कुछ उन्होंने पृथ्वी वाले ड्रग लिए थे कि भाई ये पूरा जीआई ट्रैक को, ऐसा सिस्टम को ये करता है। तो उसके रिलेटेड हमने कुछ एक जो प्रेडिक्टिव, और आर्टिकल भी पब्लिश हो चुके हैं 2-3 ऐसे। मैं आपको अभी से, ऑलरेडी टाइम बहुत हो गया इसलिए उसको डिटेल में नहीं जाएंगे, but if you're interested I will help you in the design of such activity. Okay, but again, as already I told, preclinical and clinical, even clinical trial phase 1 phase 2 phase 3, ये भी सारा predictive ही है। इसीलिए कोई भी आप गारंटी नहीं दे सकते कि जो यहाँ आया वो 100% वहां आएगा। और प्री क्लिनिकल में तो मैंने जैसे बोला कि बोटल नेक है, जब प्री क्लिनिकल से क्लिनिकल में ड्रग मूव होती है तो बहुत सारी ड्रग वैसे ही फेल हो जाती है। लेकिन आयुर्वेद में उल्टा होता है, जो रिवर्स फार्माकोलॉजी बोला, ऑलरेडी ड्रग प्रूवन है, यहाँ पे जस्ट आपको साइंटिफिक अप्रोच उसको देना है, तो उसमें 100% रिजल्ट मिलते हैं। वहां पे ड्रग फेलियर के चांसेस नहीं होते, क्योंकि ऑलरेडी ड्रग चल रहा है क्लिनिकल में, बहुत अच्छा इफेक्ट आ रहा है, उसको आपको जस्ट साइंटिफिक वे में उसको इवेल्युएट करना है। इट विल गिव वेरी गुड साइंटिफिक लिटरेचर एंड प्रोड्यूसिंग वेरी गुड एविडेंस।
Thank you, sir.
ओके, थैंक यू।
Thank you, sir. I hope our queries answered now.
So thank you.
So, thank you, sir. On the behalf of organizer, I convey our sincere thanks for delivering a wonderful session and discussing all doubts from the delegates. Thank you so much, sir, for your valuable time and experience sharing with our
Thank
delegates.
you. Thank you. Thank you.
So, all delegates, this is the end of today's session, and the tomorrow session will be delivered as scheduled time. The topic is basic biochemistry, and that will be delivered by Dr. Sanjay Bhatt, sir, Assistant Professor from the Department of Biochemistry, MDB, Automobile State Medical College, Devriya, Uttar Pradesh. So, be on time. Tomorrow session will start at 6 p.m. on basic biochemistry. Thank you everyone for joining.